Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
I told you on Monday night that I shouldn't sing, but if I was going to sing, I think I would probably learn how to rap, and I would be rapping about scaling the science. I really have a great appreciation I want to mention again Carolyn Remick, our co-chair, who has been very helpful all the way throughout our planning, and working with Jim Hunt and others who were present in our early planning meetings, we came up with this theme of scaling the science. And I'm not going to talk more about it, but I want to express my thanks for providing this major input that helped us with our program. So we'll let you introduce, Jim, what's going to happen next, and we're looking forward to it.
Thank you. Carolyn, did you want to load the next presentation? The joy of having a panel is you get multiple people doing multiple things and hopefully logically ordered. I want to put a different spin on Harold's history of how we got here. It was about a year ago. We were in Carnegie Mellon in Pittsburgh in November, and the climate was very similar. And it was really the internal intellectual climate I'm referring to, because you know what it's like outside here today. But at Pittsburgh, it was an e-research conference. And what you saw was Tony Hay wandering around, making sure everybody was having a good time. He's always done that. He continues to do that. And it's why I enjoy coming to these things. But it's obvious Tony didn't do it alone. And I found this out working with Harold, that Tony needs Harold. And Harold provides the organizational structure under which this all really functions. So when Berkeley was selected a year ago to host eResearch 2010, I thought, wow, this would be great. It's going to go well, and everybody, we just sit back and watch. Well, 6 months after Berkeley was selected, and Harold showed up at Berkeley, and a number of us got together, Carolyn Remick, Dennis Baldacchi, and myself, and Harold says, so what do we call it? And actually, there was some collective wisdom. We came up with scaling the science, and Harold wrote it down and took off, and it was great. And then 3 months later, he says, okay, now how are you going to do this? And then, fortunately, Carolyn Remick was able to ask a number of my colleagues to come forward and present themselves as part of a panel that's going to really show the application of some of the technologies you've been developing and some of the needs for new technologies, really in the area of environmental science. And so environmental science, in my opinion, really is all about scaling. Scaling in space, scaling in time. And our problem is we don't have boundaries. We have to define our boundaries, and whenever you define boundaries, you end up having to exclude some things. And you have to make sure you include the right things and you don't include the things that aren't significant. And that's really the challenge of what's going on in this area. So we have 4 presentations, 3 from the academic community and 1 from a federal regulatory agency. And it's very difficult to get all 4 of them to follow guidelines on how long they're supposed to talk. And I keep threatening that I'm taller than they all are, so I will use that advantage when necessary. So we're going to have 4 presentations. The 4 will be in sequence. And at the end of the 4th one, I will ask the presenters to join me at the table up in front, and then we will engage in some questions and answers. And this crowd has not been shy on asking questions, and so I think it'll be fun for you to see what some challenges are and where some of your technologies would apply. So with no further ado, the first speaker is Professor Garrison Sposito. He's in the Berkeley Department of Environmental Science, Policy and Management, and a co-appointment in my Department of Civil and Environmental Engineering. I met Gary about 30 years ago when I was a graduate student in Southern California. He'd been recently joined the faculty at UC Riverside. And my initial interactions with him were largely I was scared. No matter what I knew a little bit about, he knew more about. Well, fortunately, I graduated and I came up here, and then about 20 years ago, Gary came up here. And he's been leading an effort, and he hasn't reminded me of all the things I don't know too often. But he's been leading an effort in biogeochemical processes at multiple scales, from the molecular scale up to aquifer sort of scales. And he'll be providing a summary of some of that work for us now. Gary, please.
Can you?
Bill here.
There's probably a mic connection up here anyway. Can you hear it all right? Maybe I need to move it. There, is that better? Like if I do this, maybe it's better. Thank you. So I'm glad to be here. This is very interesting, what I've heard so far. Very, very fascinating. Well, let me tell you about myself, what we do. We're very interested in what we call subsurface environments. This means that we're interested in what happens in geologic formations. We're interested in what happens in sediments, what happens in compacted clay barriers, which are not exactly subsurface, but they are used to help in terms of containing contamination and the like. So that's sort of what we do. However, our program has always been based in a molecular approach to these problems, with a good amount of what we would call pore scale modeling, which means that we understand that these are media which have solid parts and pore parts, and so we need to understand the organization of the pores and the way in which they might determine what goes on. More specifically, recently we've been involved with problems that require considerations of very long time scales. The most recent problem is I'm part of the new Energy Frontier Research Center at Lawrence Berkeley Lab, which is trying to come up with innovative ways to control supercritical carbon dioxide that's injected into a geologic formation, on top of which there better be a clay capstone, which is designed to protect the world against that CO2 ever coming back up again. Now, in our approach to this problem, it has to be done over millennia in order for the CO2 to react and form a carbonate mineral, so it eventually will be a safe deposit. Now, what I want to say is our strategy here is to look for I'm going to borrow a phrase also from ecology, to look for a keystone molecular parameter or parameters without which the system probably couldn't function as we know it. That's really how we do the scaling up. We actually go the other way, is what is going on there. I'll give you an example that everybody knows about. Let's take as an example, and this is out of the physical realm. I told Jim I was going to do this. Let's take as an example Craig Venter's genome. That's probably the best sequenced, best understood human genome that we've got. It's a fully diploid genome. Now, in there you'll find strands of DNA that are for blue eyes and fair skin. You'll find strands of DNA that correspond to antisocial behavior, disorderly conduct, novelty-seeking personality, preference for day over night. There it is. Those are keystone molecular parameters that help define the person. And those of you who've heard of Craig Venter or know of his work or know him can say, yes, that's right. Well, that's exactly what we look for, just like the strands of DNA serve to identify the characteristics of the individual in the case of the human genome, or any other genome for that matter. Now, our focus has been on systems through which transport is expected to be very slow. That's why they're protective. So clay barriers come to mind, either natural or synthetic. But we also get involved with studies of paleotemperature. So we're having to go back a long time in time to figure out what the temperature was like. For thinking about paleoclimate, this is very important. Or in other cases, for thinking about the evolution of aquifers, subsurface systems that contain fluids, including water. Now, what we have focused on is to do molecular simulations of the diffusion process. And we do that with supercomputers from the National Energy Center or with appropriate clusters and so forth at LBL. And these simulations basically involve solving the Newton-Euler equations for a large number of molecules. And that means we have to know the forces And we are with those forces like Alice Waters is with her cooking. We look for the very best ingredients. Out of that, you get the very best product. So a lot of our time is just finding the very best expression of the forces, whether from quantum mechanics or from empirical work. Once we do that, we solve those equations and we just let that system evolve in time, and out of that comes the diffusion coefficients that we need. And what we have been able to do by means of that is to take that molecular information and predict continuum scale or larger scale diffusion coefficients that are very small numbers. And we've been able to do this successfully time and again for engineered clay barriers. We're working on the CO2 system now. We've also worked with the geologic formations for which people have ideas about paleotemperature and so forth. And it works really, really well. And that's because the simulations are very accurate. We run in molecular world, we run for tens and hundreds of nanoseconds, which if you know about computation and molecular scale phenomena, is a really, really long time. And so we need powerful machines. We need very, very sophisticated and clever software to do this job. The physics of this is not difficult. Newton's laws, after all. But doing the computation is tricky, and we have to think about how to make our system sufficiently sized to be able to get a relevant result and so forth. But it does work very, very well. And people have been using the results accordingly because they find that we've identified the molecular signature, because we're talking about systems over which the time evolution of concern is really long, and therefore very tiny diffusion coefficients for motions matter. One place that I want to emphasize, and then I'll stop, I think, probably close to my 5 minutes, 1/3 of a Warhol unit, right? That's all I get, is this. That is to say, we can do with molecular simulations, and many of you probably know this, we can do with molecular simulations what is often really, really hard to do experimentally with spectroscopy or any other technique. One example is the diffusion coefficients of different isotopes of the same element. That's really, really hard to measure. If you're talking about a noble gas, which is often used as a tracer in subsurface work, and we're talking about argon 36 or 40, the diffusion coefficient differences are really tiny. We can simulate those to as many decimal places as you want, however, and if our forces are accurate, we've got it right. So what we did, one of our first things to do was just find the isotopic mass dependence of diffusion coefficients. And we found that all the people in the field who had been using some sort of approach to try to figure this out, because they had to have it for their work to do the upscaling, had it wrong. Every single one of them had it wrong. And so when we put the right answers in, we got results that were amazingly good with respect to experimental data. And there's only been one group so far ever measure that mass dependence. It's really, really hard to do. So we do focus in on that as well. What proves to be really hard to measure that we might be able to do in simulation very accurately to get that molecular signature that has been determined is key to understanding the upscaled behavior. That's, in a nutshell, what we do. And computation is a huge part of it. Thank you very much.
Thank you, Gary. And Mark, if you can set up your slides. Our next speaker is Professor Mark Stacey in the Department of Civil and Environmental Engineering at UC Berkeley. Mark's been with us about 10 years, joining us from university on the other side of the bay. One of the things that I would like to tell you about Mark is his ability to transition our environmental engineering program from what was traditionally a focus on hydraulics to environmental fluid mechanics. In the area of hydraulics, it was all about scaling, but it was really about scaling a big pipe to a small pipe, an open channel of one dimension to another. How do you build a spillway? You construct it in the lab. The systems that Mark deals with are environmental fluids, whether it's an estuary, an open ocean, systems that are stratified, things that have bottoms that are not well defined because of the presence of mud, and hydrologic inputs that change flows by orders of magnitude, both in direction and timing of that. So with that, it's been fun to watch Mark evolve his research program at Berkeley, dealing with environmental resource issues, water, and ecosystem interactions. Please, Mark.
Thank you, Jim. It's a pleasure to be here. As Jim introduced me, my emphasis is on the physics or the fluid mechanics of flows in the environment. And the image here is just a surface slick from one of the systems that I work in, and you start to see already the range of scales that appear in these systems. And the scales go much beyond those illustrated here as well, and I'll talk about that in a moment. In a general sense, my approach is actually very similar to what Gary just described, but at a different set of scales, in that my goal in my research is to understand the underlying mechanisms, the underlying processes that govern the physics of these systems, and then use that understanding of the mechanisms to build up an understanding of what's happening at a larger scale. And so it's at a philosophic level very similar. I want to just give a few kind of examples of the range of scales that exist. The upper right image here is a satellite image of the surface of Monterey Bay, showing you some of the larger scale circulation. The center image is one I'm going to come back to in a few minutes, showing what I would call an intermediate scale feature, and then the small scale sort of perturbations to distributions of things at the surface is shown at the bottom. You have this enormous range of scales, from the largest scale circulations down to the smallest scale of the turbulent motions, and beyond that, even to the molecular processes. And I think that the applications that we're working on, whatever they might be, whether they're linking to the ecosystem, to individual biological processes or individuals in the environment, sediment transport and the like, that's usually going to define the scales that you're interested in. However, there are many examples where you can't focus on an individual scale, and instead you need to span a wide range of scales. And I want to give you a couple examples of those in the next couple minutes. Okay, so the first of these, let me give you a little, I guess, geographic introduction to the bay. For those not familiar, we're sitting up here next to what people typically think of San Francisco Bay is the region adjacent to San Francisco, and is really known as Central San Francisco Bay. The arm of the bay that extends to the south I'll refer to as South Bay, so from the Oakland Bay Bridge south. And then even further south, below the Dumbarton Bridge at the Dumbarton Narrows, you have what's known as the Far South Bay. Now, the reason I'm introducing you to this is that this is the site of one of the largest restoration efforts that has been undertaken in recent years. Perhaps the Gulf Coast will surpass it in coming decades, which is the recovery of salt marsh around the perimeter of San Francisco Bay. These were historically marshes that were converted into salt production, levied off, pumped out, and converted into salt ponds. For those familiar with the mosaic of different colors, that's because of different salinities in these ponds. But close to 10 years ago now, much of this property was reacquired by the government for public use and is now going to be restored to salt marsh types of habitat like are shown here. Well, that's a noble cause, but I think we need to think carefully about what the implications are for the estuary. And I want to talk a little about the scales associated with that. Ignore the words. We'll talk about the map here. This is that region of the bay that's in the far South Bay, below the Dumbarton Narrows. If you take a look at the domain of the estuary here, the estuary being the connection from where the tides enter at the mouth of the estuary to the river that enters, that scale is very similar to the scale of the restoration that's going to be undertaken. And so an open question, because restoration has never been pursued at the scale of the actual system that it's connected to, is what are the implications for the adjoining estuary when you restore at a very large scale like this? To understand that, we need to think about how restoration interacts with the estuary, and that's really a physical process, and that's sort of where I start to link into this, is what's the physical linkage between the restoration site and the bay. All right, so let's look at the scales of interest here. So this is San Francisco Bay. Perhaps you're interested in how large-scale restoration affects the ecosystem of the bay. As you zoom in on the far South Bay, we get to this scale that we were just looking at, and you see the restoration around the perimeter here. Zooming in even further, you get to the scale of these individual ponds. And then the way those ponds are connected to the estuary is through small breaches and channels that connect them. And so if you want to think about how restoration of marsh in these habitats affect this scale, you've got to start from here, think about the physical linkages, and then scale it back up to what's happening in the ecosystem at the large scale. To do that, we're taking a variety of approaches, including observations and modeling. This is a schematic of the modeling approach that we're using, where we use a full bay and, in fact, extending well into the coastal ocean numerical hydrodynamic model, solves the underlying governing equations, on this large domain extending out to the coastal ocean, but then finer and finer resolution as we approach the region of interest. And you can see here, as we zoom in on this restoration site, the high resolution that we achieve, in fact, even down to the detailed bathymetry of the linkage between the estuary and the restoration site. This requires massively parallel computing, extremely efficient both coding and computational work, as well as getting the underlying physical processes correct. And then only with that can we hope to scale back up from the detailed processes here that are where the interaction is actually happening to understand what's happening at the large scale. Okay, now I promised Jim that I'd keep it to 5 minutes, so I'm going to skip past a little bit of this, but I do want to point out this one sort of scale feature. And this is another part of South San Francisco Bay where there's a sharp transition between a deep channel and a shallow shoal region. And along that transition, we frequently see these frontal structures. And these are regions where floating debris is collected. What they're a signature of is a convergence of water masses that then downwells. And along that convergence, you have this collection of floating debris, bubbles, et cetera. Why is this connected to scales? Well, this is a feature that on its own spans a wide range of scales. It might be only a few meters across here, and there's a sharp transition in water quality characteristics across that transition. And yet it extends for kilometers along the transition of the bay here. And so just this feature on its own forces us to think about small-scale local processes as well as the larger scale mechanisms that create them and the implications for transport at the larger scale. And you can't decouple the two in this particular case. So in the first case, we have one where small-scale processes govern the larger scale interaction. Here we have an actual process or mechanism that itself spans a wide range of scales. Okay, some other implications for transport. To summarize, environmental flows in a general sense are characterized by a wide range of length scales. The applications frequently span these scales, and I didn't have a chance to talk about the observational programs. I did talk about the modeling work, but it's critical that the observational work and the modeling work that goes into understanding these systems uses the appropriate technologies to capture the right scales and span the scales, and then the models need to reflect that as well. Thank you.
Thank you, Mark. We're transitioning now to the third speaker, Professor Stephanie Carlson, the Department of Environmental Science, Policy and Management at Berkeley again. I want to do a little background. Berkeley is known for its comprehensive excellence, although it was mentioned a day and a half ago that the National Research Council ratings were a backslide in terms of how we assess things. But Berkeley did well in the ratings, so we like it. So the term comprehensive excellence isn't actually universal. It wasn't universal a few years ago. And my own biased opinion was we were lacking somebody who understood fisheries. I mean, you would think sitting on the coast we'd have somebody, and we did, but they retired and the department never hired somebody. Until 3 years ago, Stephanie said yes. And when Stephanie said yes, everybody said, oh, great. And the burden is now on her shoulders as everybody depends upon her to solve all these interesting problems that drive a lot of water resource allocation schemes. So Stephanie's our fishery biologist. He worries about coastal fishes that decide to migrate between the ocean and the coastal streams, which tries to integrate all aspects of the life cycle of these fish and teaching us all the things we needed to know. Stephanie, please.
Thanks, Jim. Can you hear me all right? Okay. Well, as Jim mentioned, I'm an ecologist. I work primarily with fishes, and in particular Pacific salmon. And my work really spans sort of fine-scale studies of the dynamics of individual populations to larger-scale studies that consider the dynamics of multiple populations that are interacting. and I'll tell you a little bit about this research today. But first, just a brief introduction to Pacific salmon. So as Jim mentioned, the fish that I work with, Pacific salmon, are migratory. So they spend the first part of their life cycle in freshwater. They breed in freshwater, spend a number of years in freshwater rearing before they migrate to the ocean, where they spend another 1 to 5 years feeding and growing before they migrate back into freshwaters to breed. And one of the hallmarks of salmon biology is that when they return to breed, they tend to return to their natal sites to breed. And this leads to the formation of reproductively isolated populations. So you might imagine that we have populations spawning in different river systems that are reproductively isolated and experiencing different conditions. So they're experiencing, for example, differences in river flow, river temperature. And these differences in local conditions acting on these reproductively isolated populations leads to the divergence of salmon populations in factors like growth rate, for example. And differences in traits among population, including in traits like growth rate, but also traits like the age at which they migrate to the ocean or the timing at which they migrate to the ocean, leads to differences in their population dynamics. And so as an example, I just wanted to show you 100 years of catch data from the Bristol Bay region in Alaska, which highlights that different population complexes have been important to the total catch at different times across this 100-year time period. So for the first 70 years of this time period, this Naknek-Kvichak stock, highlighted here in maroon, was the primary producer in this system. It was the dominant producer in terms of the catches. But in the 1970s, ocean conditions changed, and all of a sudden this Igigik stock, shown here at yellow, that was at very low levels for the first 70 years, it all of a sudden turned on, and now this Igigik stock is the primary producer in this system. And so the authors of this study argue that maintaining multiple populations that differ in their dynamics provides some insurance against future change and perturbation. And so in terms of thinking about the dynamics of individual populations versus a complex of populations, we can borrow from economics and consider the idea of the portfolio effect. So I'm sure many of you are aware of the general idea of a portfolio effect. The idea being that if you have a portfolio of financial assets and you invest in diverse assets, that will minimize the risk of the portfolio of assets. By direct analogy, in ecology, we can instead consider a portfolio of populations or species. And here the idea is that we want to manage for diverse traits, which will then generate a diversity of dynamics and create a stabilizing portfolio effect. So if we return to that work in Bristol Bay that I was just mentioning, these researchers just had a very recent paper in which they actually quantified the portfolio effect generated by the diverse dynamics that I was showing you earlier. And what they found was that the dynamics in this system, that salmon returns to this system, were 41 to 77% more stable, depending on the scale of aggregation, than the variability in individual stocks. And so this research really highlights the importance of habitat diversity in generating trait diversity, which leads to differences in their dynamics and creates this stabilizing portfolio effect. So with this general idea in mind, I want to next turn our attention to California. We have 3 species of anadromous Pacific salmon here in California, all of which are imperiled, and you'll hear a bit more about that in the next talk by Charlotte. And so what my group's work has been doing is to try and understand this bottom-up influence of habitat diversity in terms of generating trait diversity, dynamical diversity among populations, and stability of population complexes. So for example, we're conducting fine-scale studies in which we're trying to understand how habitat availability and diversity influences the dynamics of individual populations. This is a study conducted by my graduate student, Jason Juan, in the Point Reyes National Seashore, in which he's studying a stream that becomes fragmented over the summer low-flow period. And depending on the precipitation patterns in the preceding winter and spring, the amount of habitat available to the fishes can vary significantly among years. And what we're finding is that there's also considerable differences in over-summer survival rates and over-summer growth rates. So here we're really trying to understand the fine-scale effect of habitat availability and diversity in determining the dynamics of this population. But we're also considering a broader scale perspective in which we're utilizing long-term data sets. So these are data sets on the abundance of Chinook salmon in California's Central Valley. This is a stock complex that collapsed in 2008. And there are two main river basins in the Central Valley, the Sacramento River Basin and the San Joaquin Basin, both of which contain multiple populations. But we're finding that despite the fact that multiple populations or multiple stocks still exist, that the dynamics of these stocks are synchronized. And so this work is sort of emphasizing that not only do we need to maintain multiple stocks, but those stocks need to differ in their dynamics for this stabilizing portfolio effect to be realized. And so our work is really trying to span this range of fine-scale studies to understand the influence of specific habitat attributes and habitat diversity on the dynamics of individual populations, but also a broader scale consideration of how the dynamics of individual populations then interact to influence the dynamics of a complex of populations. So with that, I'll turn it over to Charlotte.
Thank you, Stephanie. Our final presentation is by Charlotte Ambrose. Charlotte works with the National Marine Fisheries Service, part of the National Oceanographic and Atmospheric Administration. She's located out of Santa Rosa, California. And we in the Berkeley Water Center have been working with Charlotte for 3 years or so, collaborating with her and learning from her and her team what the real issues are when you start looking at environmental data analysis, particularly for salmon habitat restoration. And each time I am with Charlotte, I learn new things. And most of the time, I'm learning what an impossible job that she has with the mandates of the Endangered Species Act to restore habitat for endangered species, as Charlotte will tell us about some of these species are. And the consequence of having this mandate with limited resources, limited time, and something that has to be done. And amazingly enough, she's pulled it off with a coho recovery plan that was issued a few months ago. What was amazing as she was doing all this, she was working with us thinking, what are the next steps? Because there's additional species that have to be thought through in terms of their recovering planning, and what environmental data is needed to make the decisions that have to be made. So what you saw in Stephanie's talk was sort of expanding, scaling into ecosystem complexity. And I'm going to anticipate what you're going to hear from Charlotte. Besides having this ecosystem complexity, there's also the human ecosystem complexity that comes into this. Charlotte.
Thank you, Jim. So a few months ago, I was asked to come present to Microsoft eScience. And I always had some suspicions that Jim was crazy. Then I knew it. But I realize now, and of course, you know, I was thinking to myself, I'm a typical biologist with a degree, you know, a natural resources degree from University of Georgia. I'm a federal bureaucrat. What is he thinking? But after being here, I realized that he was brilliant in inviting me, because the gap between policy and science is growing, and we need to find a place where we can start collaborating. So this is what I have felt like being here, an anomaly. And I asked Carolyn, I said, am I the only federal bureaucrat here? And she said yes. Dan Jansen looked down from a bluff. The water was like glass, the coho were in rows, and they lay there still. Every now and then one would wiggle its tail to keep its place in line. They lay there by the thousands, as far as the eye could see. Thanksgiving on the Garcia River in Central California coast coho, salmon land. Salmon coming back up to spawn. There has been a notable lack of quantitative data and information regarding the life histories of salmon and steelhead. And because of this lack of quantitative data so necessary, the Division of Fish and Game has initiated a program of study at Waddell Creek, a collapsed American economy. This is the 1930s. Shapovalov and Taft, 1954. That study is still the most comprehensive work on the life history for Central Coast California salmonids. The coho salmon that Dan Jansen saw are nearly extirpated, and the population across its range almost extinct. Central California coast coho salmon. They were listed as threatened under the Federal Endangered Species Act in 1996. They were relisted to endangered of extinction in 2005, and a recovery plan has turned to extinction prevention. We believe there's been a 99% reduction in total population. A species that has persisted in abundance for almost 1 million years in one human lifetime is faced with extinction. I am Charlotte Ambrose, and I am the recovery coordinator for the Central California coast coho. When a species is listed under the Federal Endangered Species Act, it becomes the responsibility of that federal agency to develop a plan for its recovery. There are 27 populations of salmon and steelhead listed across the Pacific Northwest. There are 10 here in California. Of the 10 populations, they're organized into what we call recovery domains. There are 4 here in California. Each recovery domain has 1 or more salmon or steelhead assigned to it. A technical recovery team led by our NOAA Science Center working on only the population aspects of the recovery plan, and a recovery coordinator responsible for facilitating the development and finalization of the plans themselves. My area of responsibility runs from Redwood Creek up in Humboldt County, south through the San Francisco Bay to Aptos Creek in Santa Cruz. I have 4 populations, 2 steelhead and 2 salmon. In general, NOAA's recovery planning process here in California involves the Science Center providing the historical population structure, the biological viability, research and monitoring recommendations. They give those technical memoranda to me. My responsibility is to herd cats. I pull in scientists, agencies, any NIMS staff, the public stakeholders, anyone who is willing to work with me to assess conditions and create these modules for these recovery plans. And of course, we're a bureaucracy, so everything has to go through that bureaucratic sieve. Are we meeting our Endangered Species Act requirements, our headquarter policies, our government accounting office audits, et cetera, et cetera, et cetera. The core components of a recovery plan involve site-specific recovery actions that improve impaired habitats, reduce threats, plan for natural events, that increase the probability of survival, abundance, spatial structure, growth rate, diversity of populations, and also a tracking system to determine whether or not we're achieving those objectives over time. These items are assessed and articulated at both a site, a stream, a watershed, and at population scales. So our population work was done by our Science Center. They modeled the historical potential of streams to support adults and juveniles. This became our universe in which we assessed habitats and threats that act on these populations. Our assessment of habitats scaled towards understanding the suitability of specific watershed conditions and functions, ecosystem processes, to support survival of specific life stages. So for example, and you got this earlier, what habitat needs are required to ensure that an egg survives in the red or in the stream, in the spawning gravels? And for that hatched egg, an alevin, to live and survive in the interstitial spaces of those gravels, to become a juvenile who lives in the stream for 2 to 3 years, freshwater systems, to outmigrate as smolts, to live in the marine environment, and then come back to their natal systems to spawn again. Salmon die after spawning, so we can't really assess those habitats, but their decomposing bodies are critical nutrients back into the system. So we identify key processes, we identify the habitats that will inform those key processes, as well as the threats acting directly on populations or indirectly through habitat impairment. We submitted a request to the public for information, and we were overwhelmed by the response. We found an informed public, we found an interested public, but we also had a whole lot of data that we didn't know what to do with. We partnered with the Nature Conservancy And we've used their process as a tool to warehouse both our quantitative data as well as our qualitative data. It's a tool where we can assess life stages, watersheds, and populations generally to get at this scorecard, which allows us to identify if there are any broad patterns across any of those scales. And from there, at least a first shot at what do we need to do where, and if asked, we can explain why. So the perfect world, site-specific data, scale to a stream reach, scale to a watershed, scale to the population itself. And then, of course, being able to answer the question, how are you, NOAA, assessing global climate change on all of these variables? Data challenges are obvious, and I think this group is fully aware of all of them, but decentralized, lacking precision, accuracy, no common terminologies. I mean, we know this. I heard this over and over and over in so many of the sessions over the last few days. But we still must make decisions. And we must make decisions at those various scales with the best information we have, and we are making broad statements about watershed processes. We are information rich, we have an interested public, but we are data poor. And while there are state efforts ongoing over the last 15 years to create an infrastructure by which we can assess, assimilate, analyze these data, test our hypotheses, that's not yet forthcoming. Scale challenges. Our understanding of fish habitat requirements are largely based on laboratory and site-specific studies that examine a single requirement for a single species at one point in its life cycle. And we have to aggregate those together to say something about ecosystem processes and their sustainability of our salmon populations. And I'm going to close here. I've got 1 or 2 slides left, Jim. By saying a few things. Agencies operate under mandated timelines. We're making decisions using the best information that we have with the time that we have and the staff that we have available to us, and that will lead you to an obvious conclusion. The low-hanging fruit is what gets picked. Us assembling, interpreting all of the information in those site-specific studies is impractical for us. We do need to communicate across disciplines, but in my work with Berkeley Water Center and Lawrence Berkeley Lab and Microsoft, there is a language barrier. And we need to get beyond those language barriers, not only between ourselves, but also the public. And funding. If any of you have read the recent Dr. Jane Goodall's book, you will have perhaps noticed that in her book there were a few who took bold moves and created a Herculean effort to prevent the extinction of a species. Then came the funding, and then came the science. I am finding hope in what's called a Datacube. It's been a pleasure being able to work with Berkeley Water Center and Microsoft, and look forward to our process that's ahead of us to work together and collaborate and face these challenges that are before us, both at a scientific level and also at that human dimension scale. And like Walt Disney said, it's kind of fun doing the impossible. Thank you, Microsoft, for inviting me. Thank you, Jim, for inviting me. That's all.
The mic, please. I would like. So there's people with the mics. You're in charge. We have a question here.
周一ワタ from University of Tokyo. Thank you very much for the excellent overview of this very important issue. And one thing I want to ask you is the differences about time constant. In case of fishermen, the time constant is 1 second to 1 day to get the fish and to sell in the market. And in the market, time constant is 1 week or within 1 week. And the ecological system is 1 generation or 1 year and 100 years or 1,000 years. And how to harmonize or how to get a comprehensive and harmonized solution by adjusting time constant by the scale of science.
Obviously, Stephanie and Charlotte should start with this one.
This is a great question, a difficult one. In terms of thinking about the time scale that fishermen are interested in, it does seem that their catch in the immediate future is obviously probably their primary concern, but also for most of the fishers along our coast, and this is true more generally along the West Coast, it's their livelihood. And so they're also interested in the long-term sustainability of these organisms, which means that they're also going to be interested in the timescales that are relevant to the dynamics of the population, which, as you mentioned, is at the generation scale. And so I think in terms of thinking about short-term economic possibilities versus a longer-term outlook, it necessarily brings those timescales together and causes them to converge, such that fishers and others that are taking advantage of this resource, that are able to capitalize on this resource, should therefore be invested in a timescale that's relevant to the dynamics of the population as well. Do you have anything to add?
No, that's beautiful.
You're okay with that, Charlotte?
Perfect, yeah.
I think this is a question for Charlotte, specifically on the salmon and steelhead populations. Do you have, or are you building, a model which tells you if suddenly somebody did give you, 10 million, some funding agency, or foundation was to make a huge investment, do you know what you do with it based on a model that encompasses the entire life cycle? Or are you at the point where you're data collecting at the moment, just trying to piece together a picture of how bad things are?
So I'm going to answer that from two sides of the equation, and that is the sequence and process that I outlined today was a process that we're using in California. The Northwest region, Oregon, Washington, Idaho, their science center are much more involved with the recovery planning process than they are here in California. And there are Paul McElhaney up in that region has done extensive modeling, and so that does exist. Those models do exist. We try to take those models and apply them down here. But our science center, we have a very bright divided line between our science center and our policy. For some very good and obvious reasons. And our attempts of using those models, because they were from Oregon and Washington, really didn't characterize the conditions and the conditions and ecosystems here in California. That said, we did attempt to hire some consultants to do some modeling work for us. We tested some of the outputs of those models with some data that we had, and found that the models themselves just were not giving us the results that we needed. The other side of this equation is that human dimension. And I was quite surprised by the level of interest that came to us when we put forth a request for data and information from the public, from stakeholders. And I had somebody tell me, I have data for over 10 years that have been sitting on my shelves and nobody has asked. So the social element in this is that if we can create a system whereby people in the public, our watershed, that hierarchy, that societal hierarchy where our watershed groups and our local governments and agencies and state and federal are partnering and working together, and we have that ownership at the ground level, at the watershed level, with people compiling some of this information that we need. For me, I think that will result in recovery.
Thank you.
I guess a question mainly for Gary. You've told us a lot about the characteristics of the sort of fluid molecules, both gas and liquid, that are flowing through the medium. What do you do about the geometry of the medium itself?
Well, there are two things that are done. First of all, in the case of movement or transport of material through a compacted barrier, for example, and maybe also some natural clay formations, it turns out that the transport factorizes into a part which represents an average over the pore geometry and a part which is molecular in nature. Now that took some time to deduce from the data that were available. So the problem turns out to be fairly simple then, because it means that the diffusion is a product of a factor which in principle can be calculated or could be deduced empirically, which characterizes the structure, the matrix structure, if you like, the pore connectivity, the variability in size, etc., and a part which has to do with the interaction between whatever is moving and the fluid in which it is moving and the nearby solid matrix. So that part is the part we can simulate. The other part we could simulate, but in fact it's easier to deal with it by just getting the information empirically. So one could do more there. I mean, one could go at higher scales and make models. People do this, as you know, I think, of these pore scale things that go on as well. But it isn't necessary to do this. What we find is the molecular signature is more important to the long time behavior that's of concern in these barriers that we're working on, because their integrity for a very long period of time is the issue. And so that's what we're trying to understand, is processes that are going on that will take a long time. Ultimately, we're interested in preventing the barrier from failing, so we need to know some things about that. So that's a kind of straightforward, simple answer to your question. It isn't so uncommon to see this factorization, actually.
Yeah, it's Kenji Takeda from Southampton. I'm involved in a project looking at aircraft design, and it's actually looking at an aircraft as a sort of system or a fleet of aircraft. So looking at how the behavior of a fleet of 1,000 aircraft behaves over 50 years, and then being able to drill down to say, okay, well, how does this, if I change this rivet, how does it then affect the overall fleet of aircraft over a long period of time? And we're building tools to help the decision-making process for the stakeholders to say, oh, well, actually, we want an airplane like this. And so it's decision support. We're building a decision support system based on multi-scale simulation and testing. And it occurred to me that it's sort of the thing that, you know, in terms of what I've seen, decision support in order to create strategies, pulling in simulation data, observational data. I mean, is that something, having a decision support tool where you can pull in all the data from all over the place in a very dynamic way as you're discussing a strategy, or saying, well, actually, we need more data over here? I mean, is that, as a premise, something that could be of interest?
I'd like Mark to start off answering that, then Charlotte to make a few comments.
Yeah, the context for some of the work that I mentioned here is exactly as you're sort of characterizing as decision support systems. And in the context of this large restoration effort, there are specific decisions being made about where to put breaches and levees, which areas to restore first, whether to bring in sediment to build up the marsh plain or allow it to handle that happen naturally. A whole bunch of little decisions that go into the overall restoration effort. What we're trying to do is to, as I think I said at the opening of my talk, is to understand and characterize the underlying dynamics so that we can ask what happens when we change the rivet as you scale it up. Along the way, there's opportunities to identify data gaps. The tools to do that quantitatively are maybe a little less accessible. And I think that's where we're sort of struggling right now. It becomes a little bit qualitative in how we think about that. But you can see it when it's missing. Defining rigorous ways to identify specific data gaps, and what decision makers really want to know is what's the most valuable data that we can go out and collect, because it all costs money. And so prioritizing data collection efforts is another thing that it'd be nice to be able to do better at, but it's a component of what we need to do.
Charlotte, did you want to? Come on, you can say
A feature of working across multiple scales is a thing called emergent scale properties, such that the sum of the pieces don't equal that of the whole. And you're all working at different systems across different ranges of scales. I'm just kind of curious, what emergent scale properties or processes have you discovered in your own work? And I think it ranges from Gary's to Mark's and Stephanie's, and et cetera.
All right, in order. Gary.
With regard to the engineered barriers or capstones and so on, so far we haven't seen it. On the other hand, I think that it's probably going to be true that there will be some emergent scale properties when it comes to the reservoir that holds supercritical CO2. That's still being looked at carefully, but the issue is whether the dynamics of the chemistry of CO2 under high temperature and pressure and a pH condition that's pretty darn low, in reaction with the system around it, particularly the mineral system, will in fact produce effects that exist at larger scales than just where that event is happening. There's a reason to think so, but this work is in a very early stage. So there are probably feedbacks between the chemistry and the transport having to do with obvious things like precipitants, plugging pores and so forth, but also there may be patterns to precipitation and so on. These are just being looked at right now at lab scales to try to understand them, to then to take that to field scale where there is some work available on this. Part of the problem here is this is taking place over millennia, at least centuries, if not millennia. So it's pretty hard to get a handle on what's going to happen in any kind of one experiment and so forth. But I have to say personally that maybe because we've been fortunate to choose systems that don't show this, we haven't had to deal with that so far in the work we've done that I just mentioned.
I think probably the short answer is I don't know, but I won't stop there. I think that the traditional approach to understanding estuarine dynamics is that you really have kind of two scales. You've got turbulent motions on scales of meters, and you've got circulations at the scale of the basin. And they interact and they feed back on one another. But I think what is starting to appear now in the work that I've been doing in recent years is an important role of intermediate scales. And it's a point where the larger scale circulations interact with the smaller scale turbulence to create features on an intermediate scale, tens of meters, that may persist for longer time scales than turbulent motions do, and are probably more important to overall transport in the system. But it's the intersection of these two, what were considered the dominant scales, that's leading these intermediate scales to be both common, persistent, and important.
Stephanie.
Well, I think maybe an example from work with fishes, at least, is related to the portfolio effect idea that I was introducing earlier. In terms of salmon biology, a lot of work is really focused at the fine scale, habitat, the influence of habitat diversity, for example, on a very local population. And it's only very recently that people have started to expand their focus beyond a particular population and try and understand how multiple populations interact and what that might mean for the stability of larger population complexes, which is actually where fisheries occur, for example. So fisheries that are harvesting in the ocean aren't focused on a single fish stock, but are actually focused on multiple stocks. And so it becomes important to understand not only the dynamics of individual populations, but how those individual populations interact to affect the stability at a larger level. And this has consequences for local economies, as I mentioned, but it also has consequences in terms of salmon evolution over the long run, because we know, for example, in terms of managing these populations, as Charlotte talked about, that they are managed at a broader scale. We're managing multiple populations within these evolutionary significant units. as a single unit. And if there's a perturbation that influences one population, there is strain among populations. So I mentioned in general that salmon are known for returning to their natal site to breed, but there is strain among populations as well. And we know this, for example, from disturbances like the eruption of Mount St. Helens. After the eruption of Mount St. Helens, several streams were decimated, and those streams eventually, when they were recreated, were populated by nearby populations. And so again, it becomes important to sort of maintain multiple populations, not only for the stabilizing effect at the complex level, but also in terms of thinking about the long-term trajectory of these populations because of this ability for salmon to stray among populations.
And Charlotte, certainly behaviors have emerged.
Well, I think I agree with my other speaker here to my left. Is we don't know, but in moving through the analysis, it was obvious that the whole was less than the sum of its parts by aggregating the way we have. And I think that as we start moving into more of those broader analysis relative to climate change, it's going to hopefully dictate more of those ecosystem process analyses and more collaboration and communication between disciplines, as we heard about from our keynote speaker the other night.
Michael?
Last question, I think.
Excellent presentation, everybody. I think this question is probably for Stephanie or, sorry, I forget everyone's names, and Charlotte. The geology, the ecology, the habitat restoration all totally makes sense, and I wonder where you would place bioengineering in, and if that's being considered at all in the efforts ongoing to not only study what's happening to the species, but how they might be augmented with the new wave of everything that's happening. You hear about things in society, and I know it's debatable, but just curious where you guys fall on this today.
For Central California Coast Coho, we have a captive broodstock, and when you say bioengineering, I'm assuming you're talking about some sort of a captive broodstock or hatchery program.
Sure, or something done in a lab if the technology presents itself. I think I heard something in the press about engineered salmon that might be introduced to help speed up the breeding process, and I don't know if that's the same thing.
Yeah, I can't make an official statement to that end.
What's your personal opinion?
When the camera's off. But I can say that the position is that if we create more suitable habitats and create more functional ecosystems, it's just from a financial perspective, they take care of themselves. And the ocean environment provides for those food resources that provide the omegas that make them such a healthy food source for us. So our first is conservative. Let's take the money, use the funds, identify what types of restoration projects, what kinds of changes in our own actions we can take to provide for the salmon and the salmon habitats first. We do have captive broodstock programs, one on the Russian River. It's a wild broodstock program. It's a collaborative process through multi-agencies and our science center where they're looking at the genetic variables to ensure that the interbreeding is reduced. And they're seeing some success now after about 11 years of that work where we have broodstock actually returning. And we are anticipating to probably extend that broodstock program to our Lagunitas and some of our streams south of San Francisco.
So just to follow up on Charlotte's comment there, I guess two ways that bioengineering has come into play, as she mentioned, is first in terms of the broodstock programs that are happening, and these are really meant to keep wild populations that are at the brink of extinction from going extinct. That's the goal of those. But the second that you mentioned is genetic engineering that's happening, and that's not really happening for our wild stocks, but rather that's focused on farm stocks. And so there are few regions of the world where we have an extensive amount of salmon farming. Those tend to be regions with a lot of fjords where you have protected bays and you can have large net pens, so around Norway, British Columbia, and off the coast of Chile. And right now, there's a very large operation off the coast of British Columbia that uses Atlantic salmon. So Atlantic salmon, as the name implies, are native to the Atlantic Ocean, not to the Pacific Ocean, but they were brought to Atlantic salmon from the Atlantic to be raised in net pens off the coast of British Columbia. And we're told that those individuals would not be able to breed because through genetic engineering, but actually individuals that have escaped those farming operations have now established self-sustaining runs off the coast of British Columbia and are apparently able to breed. So there are dangers associated with genetic engineering of these organisms, and the consequence, of course, is that there are wild stocks in those regions, and now they are interacting not only with their native organisms, but also Atlantic salmon that could potentially be competing with them and preying on them.
With that, I would like to close the panel and allow us to thank the panel one last time in unison. And you know now why I like being at Berkeley. They're just nice people around. And so we'll get away from the table here, and Tony has the floor.
I was actually going to ask a question of the panel, but don't worry.
No, you didn't get a chance.
So, switch it on. Thank you very much. It was a fascinating panel, and I was going to ask that I was surprised. I'm not fine. It's my cell phone floating around somewhere. At least this isn't an iPhone. I was very interested to see that the Datacube was being used by the Swedish Environment Agency and by NOAA, and I wondered if there was more that should be done and could be done, but we can talk about that afterwards, because talking to the SEI, it's the need to put more data, make it available, so maybe there are things we could do. Anyway, my job is to make some closing remarks and to thank you all for coming. One thing that occurred to me, and it was a no-brainer, and I'm sorry I didn't think of it before, we clearly should make this digitally available and our back numbers too, and give them a DOI and find a suitable place to host it, perhaps on our website, but perhaps there's a better venue to do that. So I'll take advice from our scholarly communications director and others as to how we make this available. So it's referenceable, you can refer to papers and get some credit. And also what I hope to do is persuade Caroline and Harold. I was talking to Jeffrey Fox, the editor of Concurrency and Computation, Practice and Experience, and he's agreed to the possibility of a special issue of the selected papers from this conference. And so if I can persuade Carolyn and Harold to act as the editors for that special issue, then they can select papers, and we need to see if we can get it out on a reasonably timely scale. So I hope that will be welcome, but since I hadn't asked Carolyn, I'm not committing to that, right? Could I just quickly ask Bill Howe to say a few words about a conference that Microsoft and the Moore Foundation are sponsoring? There's a microphone somewhere.
Thanks, Tony. I'll just keep it brief. There's flyers outside where all the other materials are, but it's the Scientific and Statistical Database Management Conference. This is the 23rd year of it. It's actually a very old conference, but we're trying to sort of raise the profile because of all the interest in eScience and all the interest in big data and so forth. And I'm a registration chair, and as Tony mentioned, Microsoft Research has kindly sponsored it, as well as the Moore Foundation. And the most important thing is it's in Portland, Oregon, so it's very accessible for probably anybody that came here, unless you came from the UK, I suppose. But anyway, so it's next summer, July 20th, 21st, and the abstract deadline is, if I remember correctly, is January 31st, 2011, so there's still plenty of time. And we're sort of all interested in all the areas that we've talked about here at eScience. And one thing I think is especially important is a little bit more emphasis on applications, which I think is something that's wonderful about this workshop. It's something that the PC has been explicitly instructed to look for. So encourage you all to submit papers. Thanks.
Thank you, Bill. No, it's a database conference which is trying to change to become more relevant to applications, and I think it's really particularly relevant for this community. So something you might like to think about. The most important thing I have to do is to give some thanks. A lot of people made this possible. I just stand at the front and buy drinks and things like that. I sometimes feel I drink for Microsoft. All right. But I should thank the event team, Melissa Kelly, Jennifer Carlson, and Sarah Dangle. You saw them outside. They were working very hard. So can we give them a round of applause? And then there's the ever-patient video team, Jeremy Crawford, Hugh Beery, William Batts, Chris Cruz, and Brian Unger. They've done a great job, and you'll be able to see some of the talks you missed in the parallel sessions because they'll be online. So can we give them a round? Thanks. And then there's the media team. So we've thank you, guys. I'm sorry you missed your round of applause.
They're back.
But the media team, Heather Mitchell, Alyssa Felder, and Kelly Berschauer. And what happened, they arranged various coordinated activities with the blogs, with the website, and also some video interviews, which I think you'll enjoy, of the 3 Jim Gray Award winners, Carol going on about her shoes, I seem to remember, and various other things. So you I hope, will enjoy those videos. Roman Stitzar has been our photographer, so thank you, Roman. So let's give the media team and the photographer a round of applause. Roman. And then there's the team that did all the planning behind the scenes, and that's, of course, Carolyn Remick, Catherine Van Ingen, Jim Hunt, and Dennis Baldocchi. They were really key to making this a success. It's a great venue, I hope you appreciate. The Claremont is a famous hotel and a wonderful place. And Carolyn carefully arranged with the Met Office to give you good weather for this week of this. So it was really great. So thank you very much to the Berkeley team for all your help. It was great. And don't forget the tutorials, it says here. I'd like to thank Mike Ziskowski for actually taking on the organization of the tutorials. He was there, but anyway, can we give him a round of applause anyway? And I think that's about it. Oh, there is one other person I should thank. Harold Javid, who without this, nothing would have happened. So I really would like us to give a special round of applause to Harold. He's been invaluable. I haven't had to worry. Things happened. I'm very grateful to Harold. Harold, thanks very much indeed. So next year, we're thinking about the exact format and venue. So if you've got suggestions, the questionnaire, you can send them. As Harold said, it'll be less marking numbers, but actually perhaps asking for your thoughts. We're thinking of various possibilities, and we'd like to hear your views. All right. So don't forget the tutorials. And there is some good news. When I last was informed that 18 of the 33 Chilean miners have now been rescued. Has that gone up or not? Okay, so that's really pretty good. Anyway, last thanks is to you guys for coming and making this, for me, a very interesting event. Slightly frustrating that there were parallel sessions I wanted to attend and I wasn't able to go to them all, but it really has been good. And I think it's an interesting community. It isn't quite like other conferences, but the question is, do we keep it the same? Do we grow it? I don't know, and it really depends on you. So you have to get something out of it, and I think having some publications you can refer to can help. But what other things could we be doing, and where should we be going with the community? How can we help the community grow and become more relevant? So thank you all again, and I hope we see you next year. Thanks a lot. I think it's lunch and tutorials, right? Thanks a lot.